Hi, good morning. Welcome to the second session. I'm Sarah Haecker. I'm the Director of the Technology section at the Alliance for Regenerative Medicine. I'm also the Chief Scientific Officer for Adjuvant Partners. This morning, we focused this panel on platform technologies. And as many of you know, at the Alliance, we have 3 technology sections now, a cell therapy section, a gene and gene-modified cell therapy section, and a tissue engineering and biomaterials section. So the purpose of this session this morning was really to explore how a couple of our companies are commercializing their platform technologies. One is a biomaterials platform currently being commercialized for research use purposes and then into the therapeutic realm. And the second is a cell therapy-based platform that is being commercialized in a number of therapeutic areas. So we're interested in understanding how these companies are exploiting these platforms and leveraging the strength of these unique assets. It is my pleasure to welcome Eduardo Bravo, our first speaker. As many of you all know, too, we've recently merged the Alliance for Regenerative Medicine with our European sister counterpart, the Alliance for Advanced Therapies. Eduardo is the chair of that organization, and we are thrilled to have this organization merged into one now. So, Eduardo.
Thank you, and thank all of you for staying. It's a difficult choice between the coffee after the first session. Let me start by saying that I'm very depressed after the previous session. You've heard how difficult it is. But I'm not depressed for that. I'm depressed because most of the examples, as I'm going to show you, probably are taken from our own experience in developing these cell therapy products. So let me explain what we do and what has happened. By the way, we're a public company, and I always make plenty of forward-looking statements. So this is what we have today, and we started, and I'm not going to cover that, with ChondroSelect, the one at the bottom. This is the first ever advanced therapy product to be approved in Europe for a medical use after the new legislation came into place. That approval happened in 2009. Great. You get an approved product, so what next? You need to get reimbursement. In Europe, that means dealing with 27 countries in some of the countries, and I'm Spanish, in Spain, then you deal with 17 autonomous regions. Once you manage to get through the 17 autonomous regions, then you deal with the hospitals. So 2 years after approval, we got the first reimbursement, being a Belgian company, of course, in Belgium. We bought we built our manufacturing facility in the Netherlands. Second approval of reimbursement, the Netherlands. We merged with a Spanish company, third reimbursement, Spain. So just to show that this is just economic data, nothing to do with politics in Europe. Despite that, once you get the product approved and reimbursed, it becomes very difficult to commercialize. We didn't have the resources, so we've just licensed it out to Swedish Orphan. So I'm not going to talk anymore about this product, which is manufactured by a CMO. It's commercialized by another company. We just take a royalty on the compound. All our efforts today is built on the development of this allogeneic adipose-derived stem cell platform of products that we are developing both in the US and in Europe for very high unmet medical need diseases. And I hate to say this, unmet medical need. Everyone talks about unmet medical needs, but I will try to explain why we're doing it. I'm not a scientist. You all know how these cells work. The other day in a paper, I read for the first time something that I could explain not being a scientist, which is these cells are Swiss Army knife products. And I truly believe that's what we've been trying to explain and successfully to investors, that these cells have this ability, as Gil was mentioning on the previous panel, of dealing and adapting the response to whatever they find, depending on how you administer the cells. What we've demonstrated is that the key mechanism of action, at least of the adipose-derived stem cells, is the control of inflammation and the interaction with the immune system. And I will go into then what are the indications that we've decided to move forward with following that mechanism of action. And then, let me tell you, why adipose? And we have the nice story. The nice story is we've chosen them because they are easier to get, plenty of fat around. I'm not talking here, I'm thinking the world. Very easy to get donors. You just go to a liposuction clinic, you ask for donors, you get 110% because the nurse volunteers as well if you're paying for the liposuction. So it's very easy, plenty of fat around. Getting the right donor is important. But they are more stable on expansion. They expand quicker than bone marrow cells. So we believe that there's good evidence that shows that adipose may be a very good source. We've also demonstrated that they are less immunogenic, so that they may be better suited for allogeneic use. Then we went in the development of one single compound, and it was a local injection of the cells, and I will go in a minute into that. But unfortunately, because of the regulators, we end up doing a huge clinical program to show also on the CMC part that, these cells were safe. So we ended up doing, 5 routes of administration, 5 animal models. So we wanted only local. We ended, with intraperitoneal, intralymphatic, intravenous administration, just to show where the cells go, how safe they were. We ended up with quality. And once you've cleared your quality, you've cleared your quality for the cells for any use. We also managed to get our manufacturing sorted out, sort of. You've heard how difficult this will be, but today we have a reliable, we've got approval, at least in Europe, for a purity, potency, and identity assay, so we can release the cells. And we have, a way of producing, thousands of doses out of each donor. So still a lot of controversy on what will happen when you choose donors and how you demonstrate consistency. But that will be the next barrier to jump. And then the question came on the IP. We were doing our best to try to protect ourselves. I still think that the whole space is not easy to protect from a pure and typical IP way of protecting the product. And at the end, almost anyone can come with a completely different cell type and come into the space. So we're just making sure through FTOs that we are not stepping into anyone's IP and trying to do our best to try to protect as much as possible the work that we do. So with that, the real truth. This is how tough has been to develop one product out of that platform. And all started with an autologous product, what we call CX401. And we licensed that compound in, let me see if I get it. We licensed that compound in 2004 after, one doctor came with very remarkable data out of 5 patients. This is what we were mentioning before about, getting some data from hospitals. And at that time, believe it or not, the decision of going into this disease area of complex perianal fistula, and you can imagine now after breakfast how nice this disease is, that it's at that time, we thought that the cells will home, will grow, and will close the fistula. That was the mechanism of action that was thought to be true when we licensed the compound, not only by us, but by the field in general. So we started our own phase 2 clinical trial, randomized, blinded trial, 50 patients, got great results. And the results were so good, that we thought that we can do almost anything, cells will work. So we rushed into a phase 3. You've heard it, big mistake. So you get your phase 2 data, then you think carefully, what is the best thing that you can do with your phase 3? So we rushed into a phase 3 in non-Crohn's fistula just to find out that in those patients you get a huge placebo response. So we couldn't find the difference that we saw in the phase 2. So at the same time, we started developing an allogeneic compound. Autologous was a pain, and it's very difficult to make a successful product for an indication where you're not curing lives, basically, and make the cost of goods be sufficient to pay for the price that you're going to be able to charge. So we started with the development of the allogeneic, and we did a small second mistake. If you do a phase 2, you need to do a real phase 2 that will answer the question. But we thought that, we were here going to be successful. So we did a phase 2 just to show that allogeneic safety was nice and that, efficacy was going to be comparable. So we did a non-comparative phase 2 trial. Efficacy was great, but of course now we don't have a placebo arm to compare with. So, some people can say this is rubbish. We don't agree, but this may be the case. So we did at the same time an investigator-initiated trial. I think that that's something that I will encourage people to do. I think that combining your work with work that is done free of charge even in small patient populations, as it was mentioned as well in the panel, is something that is really worth doing. And finally, we agree with the EMA, despite the non-comparative trial of the phase 2, on a design of a single phase 3 clinical trial that, if positive, may allow us to file for approval. So after, more than 10 years, actually 14 years of work, hopefully by June of next year, we will have the answer on whether the cells do work in this patient population, and this is a trial powered to show statistical significance with a p-value of 0.025. And I'm not going to go into more detail because that's actually the purpose of my talk later today when I present the company. Let me now go into, okay, that's the local. So that was the only thing that we were doing. But we also had two very interesting publications of work done by our own at our own lab. One was in severe sepsis, in which we demonstrated, and that's from 2009 actually, that the cells had a very pronounced effect in reducing mortality in sepsis animal models. It's if you look at it, is dose dependent, so the higher the number of cells, the more you control mortality, and more importantly, we've done it with human cells and with mouse cells, the effect is the same, and the effect is extremely pronounced. And at that time, for the first time, we mentioned that the cells had an antimicrobial effect. People laughed at us, and we didn't have an explanation of why the cells had that effect. Today, there is a scientific explanation. So again, sometimes the findings go before the science. And on top of that, we had also some very interesting data in a rheumatoid arthritis model, in which we saw again this anti-inflammatory effect of the cells in controlling the inflammation associated with the cells, and I'm not going to go into the data, but it's to show that the cells do have the ability to reduce the production of pro-inflammatory cytokines and induce the production of anti-inflammatory cytokines, IL-10. So what we've done then next is, okay, let's try, and we went into a single phase 2 clinical trial in RA patients. We wanted to demonstrate that the cells did have an effect, so RA was chosen. Why? Because on the clinical endpoints of RA, there's no discussion. So let's find a disease area in which there would be no discussion. We went for RA. Regulators asked us to wait for patients that have failed 2 biologics. And what we ended up with, unfortunately, were patients that had failed, sorry, I went one too far. So patients that have failed, 3 non-biological DMARDs, followed by 3 biologics. More than 13 years of therapy without success. Those were the patients that, were given to us, thanks. And we were giving them just 3 injections of the cells, days 1, 8, and 15, and then follow them for 6 months. Do the cells have an effect, and do the cells keep the effect, at least for 6 months? And of course, for safety, very nice side effect profile. Again, we don't have the time to go there. Let me go into the efficacy data, and let me go into EULAR, because after 15 years of failing, ACR is no longer the right thing to look at. But if you look at the EULAR criteria, 40% of the patients were still good to moderate responders 3 months after receiving just 3 injections of the cells. 24% still good to moderate responders 6 months. More impressively, 10% of the patients, and it's a group of 46, so it's a small group, but it's not 5 patients, were on remission 6 months after receiving just 3 injections of the cells, after failing for 13 years, 3 non-biological DMARDs and 3 biologics. So again, small patient population, it was not powered for efficacy, it was just a proof of activity. So now what we're doing with that, with those cells, is moving into exactly those 2 indications. One is going for remission in early RA, and the second one is we're going after the severe sepsis patients. And that's how, from let's close these tubes in the fistulas, the cells will home, grow, and close the fistula into, this is a very potent anti-inflammatory agent capable of not only controlling inflammation locally administered, that's the fistulas, but also, maybe controlling some of the very severe inflammatory diseases, RA and severe sepsis. It's just 15 years and a lot of money. So good luck going forward, and be patient and persistent. Thank you.
Thank you, Eduardo. For our second speaker, I'd like to introduce David Lightbody, the CEO of Biogelx, and he's going to be discussing a biomaterials platform. Thank you,
Good morning, and thanks for the opportunity to come along here today. We're very early into this game, and certainly very early into the regen med game, and we are fundamentally biomaterials people. We don't do any cell work ourselves. We are all very innocent chemists and work very closely with cell biologists to take our products forward. But we are fundamentally chemists, and we get into this game really not looking at regen med, but we set up this business, and we've only been trading for 18 months. The technology's been around. Well, the first patent application was 2007, 2008. I got involved about 3-4 years ago prior to trading, and we set up some commercial or pre-commercial arrangements, put a couple of contracts in place, one in South Korea and one in the UK, and that gave us the confidence that we could set up a trading entity, but it was very much geared at the 3D cell culture market, which is obviously a growing area and something that we felt we could play very sensibly in. The technology itself is based on very simple peptides, dipeptides and tripeptides. We use fundamental gelators, which are very simple hydrophobic materials or peptides that we're able to self-assemble using and that's our kind of core technology. How do we trigger self-assembly of these little peptides into nanofibers? As I mentioned, they're hydrophobic, and although they self-assemble beautifully, and we'll get some lovely systems in place, cells tend to find them a little bit difficult. So the key technology is taking surfactant materials and peptides that we can co-assemble with those nanofibers to create systems that are attractive for cell culture. And that's kind of it. So we've got our hydrophobic gelators coated with surfactants, and we have this coated fiber. Now, what we do is we modify those systems to match cell types, and that's kind of our key, is that we use different chemistries and different coating mechanisms to suit specific cell types. We also have great control over fiber density and fiber thickness that gives us wonderful control over stiffness. So we're changing physical properties, and we're changing chemical properties. And the thing that is kind of, as we'll get to, the thing that's pulled us into the regen med side is this feature, in that we make these nanofibers. We've got them 1% solution in water, and they're not assembled at that point, and they're not in a gel. The gelation takes place when they're in the presence of certain cations, which are present in cell culture media. So we supply the material in kind of a free-flowing liquid form. It sees cell culture media and cross-links into the gel, which is the obvious mechanism for putting cells into 3D culture. So that's it can be used in 2D as well, but the neat factor is that cell biologists can then examine their cells in a specific 3D matrix. And very cartoony, but this has been of great interest to a number of cell biology researchers in a number of labs, companies, and increasingly within a couple of big pharma clients, where we can take our gels and modify the stiffness from kind of the stiffness of brain up to bone. So as I mentioned, we only started trading last year, March of last year. We raised a couple of $100,000 seed funding. We did $300,000 in our first year, and rather surprisingly, I would guess, for a company of our kind of nature in this field, we did break even. As I mentioned, we've got 7 employees and 4 Ph.D. chemists. What happened at the back end of last year, we went out and we participated in a couple of awards, which we won, and this is where the company kind of transitioned and I guess is where we are today, in that we won a Royal Society of Chemistry Award in London, which was sponsored by GSK. And that got us a lot of visibility in terms of YouTube visibility, media coverage, and recognition within a couple of big pharma companies. Because the question that was asked by researchers, SMEs, and larger entities was, could this be used as an injectable? I think they saw the application before we did, in terms of using our system to deliver cells and do it in a way that the cells would then be in a fixed location because gelation would then take place in situ. The stiffness feature that I mentioned before, that's been used by a number of organizations for control of differentiation. So for stem cells, and particularly the adipose-derived MSCs, respond particularly well in terms of varying stiffness and varying their differentiation characteristics. So suddenly we're in the regen med market, and we feel at the moment as if we've got a bit of a tiger by the tail. We've got collaborative partners in place looking at small bone repair, cartilage repair, and we've got some skin R&D programs on the go at the moment. We've also been dragged into islet transplantation, using our gels to protect islets. And surprisingly to us, we have a big pharma program on at the moment using the system for drug delivery, so no cells involved, simply for small molecule delivery. So that's where we are. We've, as I say, we've been on the go for 18 months. We're now or heading into the field of regen med, and we're faced with a number of challenges and next steps. And for us, I mean, 7 chemists working. You may have gathered we're based in Scotland, between Glasgow and Edinburgh, but we're never going to go to GMP. So we're talking to partners in Europe and the US about establishing GMP for our peptides and for our final product. During this whole thing, there was an academic behind all of this who was headhunted 6 months ago, and he's now based in New York in a nanoscience center in Upper Manhattan, and we're looking to put an office and a laboratory in there over the next 6 months. As I mentioned, we are chemists. We don't do any cell biology, and we've been very clear from day one that we will not kind of cross that boundary and get into areas where we know we have limited knowledge, and we wouldn't want to get cross-threaded with our customers, but we're open to new strategic partnerships as they come along. And our challenge as a small biomaterials company, as I say, with a bit of a tiger by the tail, is we're out looking for cash and for more people. And thanks for your time.
Thank you. I think we have time for just a couple of questions, and so if there are any in the audience.
I'm going to assume this is on. For Biogelx, do you have a sort of like a set of linear dimensions that you have to work with? Would you get the same kind of gelation properties for a very large sort of spinal cord-like entity that you tried to assemble with cells versus say like islets, something on the order of 1/10 of a millimeter or so in dimension? I mean, is like they're like kind of like the production is it varying over a linear scale?
I would say that was why when we talked about bone repair, we've been very careful to keep it into small dimensions at the moment, and that's just in terms of what we understand our materials can do. We've been asked about these larger applications, and that would require additional scaffolding, we assume, alongside the gels. As I mentioned, these things are self-assembled, so the physical properties, I mean, they're pretty loose structures. But we're looking at incorporating our material possibly with other, maybe more polymeric systems, and use it as a kind of a dual system.
I was also curious as to the experimenting with like a heterogeneity, where you have like a very stiff matrix and then sort of like a very sort of much lower viscosity kind of interstitial space where the cells would flow. I was just wondering, getting experimenting with kind of a heterogeneity structure.
No, that's very early stage just now. No, really we've been focused just on our materials and varying the stiff. We can vary the stiffness characteristics from very soft up to very rigid, just using our material and varying the fiber size and fiber density.
How about porosity and hydraulic permeability?
Well, it's kind of like tissue, I mean, it's similar.
I have a question for Eduardo. So you mentioned using investigator-initiated that was part of the program. My question is really, at what point or what were the drivers behind going into RA and sepsis? So at what point did you decide the platform could be expanded there? Because it seemed to be done internally.
That was actually decided before we started the RA clinical trial. So once we started the allogeneic development and we got the approval of the European phase 3 clinical trial, then the board decided that, we could then expand the platform and try to find other indications. And the idea was, let's find one indication that will allow us to have this sort of proof of activity. We didn't have, phase 1 data, so we ended up doing this phase 1/2 clinical trial in a very severe patient population. And the idea was, we've chosen RA at that time because it was the best of the autoimmune indications where we believe that there's less controversy on the clinical endpoints. So that's the reason why we ended up there as a proof of activity.
I guess I have another one too. Then, many of the companies in developing technologies are obviously have platform capabilities of these. Some will go the route of developing or commercializing a research use only application first to potentially fund further therapeutic development. Can you comment on the rationale or the route that you got to in terms of going that way? And you mentioned looking for partnerships. How aggressively are you doing that? And in what areas have you decided to go into, or is it sort of first come, first serve?
We set up this business because we saw an opportunity in the research side, and we funded it on that basis. And that is still the core of our business. I mean, we won't let that go because we know we can do it kind of in a profitable way. And we've got customers in South Korea, a couple in the States, and throughout Europe, Italy, Spain, Portugal. We also have significant interest from big pharma using our platform for drug discovery and toxicology work. So we see that as a massive opportunity. The Regen Med thing came along because people asked us about it. We could see it was, from our point of view, high risk. It wasn't something that we had any great capability in. And we're really responding to customers. In terms of, are we out targeting people to do that? No, we're trying to respond to the wave of information that we've got coming through at the moment. I'd like to say we're less opportunistic than that, but that's probably not the case at this particular stage. What we're trying to do is get ourselves into a position that we have enough significant strategic partners in place so we can develop a plan that we can get funded. And that's where we are today.
I'd like to thank both of our speakers, and we're now on break, so thank you.